who, for the newcomers to this lecture series, he's a member of the National Academy of Sciences of Romania, and he's giving us a series of lectures. So today he was giving two lectures, one on the biological cell and one on languages with infinite words. Professor Marcus.
Thank you. This is for me a special lecture for at least two reasons. One is that it is very near by its topic to the center of computational molecular biology, which invited me here. And second reason, it is related also to my personal biography. So this is for me an opportunity to express my admiration for this center of computational molecular biology, which is now in attention of many specialists in the field because it was directly involved in the huge human genome project realized here in the United States. I will begin by telling you something at that board. I observed that they did no longer put me the...
Yeah, that's right. It's a difference.
What I tell you now happened just 40 years ago. In 1968, I met Professor Zdzisław Pawlak from Warsaw, and he just a few years ago, before our meeting then, he published a book in Polish language called Gramatyka I Matematyka. And as you can understand this title, despite the fact that you don't speak Polish language, I understood too, and I realized that I can follow many things in the book. The book was an introduction to mathematical linguistics, but one chapter caught my attention. And this chapter had the title I translate it in English now. The grammar of protein. The book was published in 1965. If I remember well, nobody at that time in the field of computer science or mathematics spoke about such a thing, grammar of protein. And I followed with great interest. It was no difficulty to follow it because it was full of pictures and of formulas. And the main idea there was to represent one amino acid by a triangle. And by associating to each side digits from 0 to 3. But with the following rule. If I put here I, here G and GK, then I should have J smaller than I, larger or equal to K. And motivation. He said, I have 20 relevant types of amino acids in the genetic code. But I have 64 ways to arrange the elements of the alphabet, 4 elements in the alphabet, in the genetic alphabet, adenine, cytosine, guanine, and another. And I have to introduce a rule to eliminate from these 20, from these 64 possibilities, to eliminate many of them in order to keep only 20 of them. And you can check that if you follow this rule with I, J, K in this alphabet, you get just 20 types, 20 possible types of such triangles. And why triangles? Clearly because by the genetic dictionary, amino acids correspond to codons, and codons are obligatory of length 3. You know that it was a moment immediately after the great discoveries in respect to the genetic code towards the middle of the past century. And we learned all these things. And then he defined a recurrent process. As a matter of fact, it corresponds to what in linguistics is called a dependency grammars, usually represented by trees. And here, for instance, the rule was any of these 20 triangles the problem is to simulate, to define a concatenation process of this triangle which simulates the protein formation. And first axiom was that all these 20 types of triangles are proteins of length 1. Then he defined a recursive rule giving the conditions to concatenate. And they can be represented. If I represent this triangle in this way, then I can concatenate new triangles only on this is the base, and this is the left side, this is the right side. I can concatenate only to the left and right side, not to the base. And then you can get various, till you reach, when you reach 0, you no longer can continue. So you can, in this way, you can get such formal proteins may have any possible finite length. And I liked very much this exercise. Also, it was not clear for me to what extent it has biological relevance. But 2 years later, 3 years later, I attended in 1971 the Linguistic Institute of America, Buffalo, New York, July, August. And I used this opportunity to organize a seminar of mathematical computational genetics. Among the, I had participants, biologists, or among participants it was, for instance, Bernard Vauquin, who is one of the authors of Algol, the first mathematical programming languages. Bernard Vauquin. And the result all these interactions gave me the opportunity to write a paper called Linguistic Structures and Generative Devices in Molecular Genetics, published in '74. And I have to tell you that at that time, in the 50s, in the 60s, main interest in computer science was toward the nervous system, all kinds of automata simulating nervous activity. But not yet such things, grammar of protein, began to come in attention only in the 80s and mainly in the 90s. And at that time, for instance, in my paper in '74, there is a bibliography of about 50 titles showing the great interest paid by linguists at that time to problems of molecular genetics. Because historically it was a very important moment. I have to tell you that beginning already in the 19th century, the field of historical linguistics was basically influenced by the evolutionary ideas promoted by Darwinian biology. So after a long period in which biology inspired linguistics, it came the moment for linguistics to pay his duty, and we began to have linguistics inspiring biology. And one of the protagonists of this movement was just Cambridge, MIT, the linguist Roman Jakobson. This was, Cambridge, MIT was the center, the World Linguistic Center, with Jakobson and Chomsky, both in the same place. But Chomsky, despite the fact that he produced formal grammars, he paid no interest to apply these formal grammars to biology. And I was obliged to make this introduction because, as you will see immediately, new generations forgot very, it seems to me they don't pay attention too much to history and to the past, and they begin things like they are the first dealing with the respective problems. And you will see immediately what is today the relation between linguistics and molecular biology. There is a field in linguistics called genetic linguistics or linguistic genetics. No, very much developed, but where the bibliography begins with the last decade of the past century. All these things were forgotten. First of all, in order to show you, because I put the title, The Biological Cell in Spectacle, you will see that this spectacle, in some respect, is impressive, in some other respect, we could say depressive. There is what is opposite. In some other respect, it is very sad and giving very many reasons to worry. Look, first, what we read in Encyclopedia of Computer Science, Anthony Ralston, editor, Oxford, in 2000, year 2000. Timeline of significant computing milestones. Look here. $500 million before Christ, first biological cell. But in another source, we read 2.5 billion years old, creates the most complex unicellular organism on the earth. How can precede this the first biological cell? Obviously, is not consistent. I have to tell you that I checked all the other sources. I don't have them here. In other sources, I found more than I found 2 billions. 2 billions ago, the first biological cell. So in this respect, it is clear that we are in a field of speculation where things can very difficult be checked. So these are for them the main important data from 200 million years before Christ, cell ascent into first memory. They moved to Conway's game of life. Why not to cellular automata? Because this is only a particular way. And then to 93, Leonard Adleman demonstrate DNA computing. Here are a few dreams of computer scientists. Look, Stephen Wolfram, 2002. Life is not unusual, no essential difference between a living being and even a human being, and a universal Turing machine. This is an extremist view, because there are other scholars who oppose completely to this. DNA is another one, Chaikin. DNA is essentially a digital software. Human beings have much more DNA than viruses and bacteria. We are universal Turing machines, and we are surrounded by universal Turing machines, but these machines differ in their program size complexity. Life is a collection of universal Turing machines whose software evolves in complexity. And then we have a convergence of theoretical physics, theoretical computer science, theoretical biology, and there's a hidden umbrella of semiotics. You will see me there. And here I move to what is claimed in the field of biosemiotics. The emergence of computational biosemiotics will continue in the new decades, facing difficult problems. Genomics remains a great challenge. This is also related to the status of identity. The identity of a biological cell is not, and not primarily, its material identity. Because we can see also for the human body identity that it is not its material identity the most important, also it is the most visible. I will not insist now on this. So, when we speak about the biological cell, we have to look at its structural identity and at its interactive identity, the way it interacts with the surrounding. So, it is true here that you cannot understand identity of a biological cell if you don't understand its alterity. But now, I will give you the absurd scenario existing at this moment in the field of biological cell. Imagine the existence of a set, A, of fields of research such that, A, they have a common object of investigation. However, the questions they raise are different. The communication between them is very weak, if not absent. One can assume that some of them ignore each other. None of them is aware of the existence of the other. Beyond a basic common terminology, each of them develops its own jargon. The journals where their results are published are almost entirely different for any two different fields in this set. And in most bibliographic references, in most fields we are considering, the year is not before 1990 and even not before 2000. And the example we have in view is just the biological cell. Because all fields whose list I will give now, partial list, because the list can be enriched with all the fields you will see now. I will give a list of 15 directions from which biological cell is approached. Each of them begins with this claim: our aim is to understand the functioning of the biological self. And after asserting this aim, each of them takes its own way, its own jargon, its own bibliography, its own journals, no dialogue between them, and this is the life in the world of science today. Not only in the world of science, because you will see, among these fields are also fields from humanities, not only from heavy science. I wrote here, Station reminds a mythological tale about an elephant and 7 blind people trying to perceive the elephant, and each of them is confusing the elephant with the part of its body where it is. And his perception is a completely distorted image from what really the elephant is. Which are the 15 fields I collected? But I repeat, I am sure each of you can add some new fields because nobody has such a rich information to cover. You know, you have first the biological approach, what biologists are doing, but I have to tell you that I observed that even among biologists, biology is now a continent where almost nobody knows what happens in a whole continent. I observed that, I checked that many biologists ignore the names of some very important biologists, but who work in another way than them. The biological approach, I leave it aside. This means the approach by people who have papers to prove they are biologists, they have a PhD in biology, or I don't know, as a matter of fact, what could be today the definition of art. Then, the linguistic approach. You see, you can understand after I told you this, that I was very interested in this approach. The linguistic approach is related to my biography. And now I put there a name. There is a great encyclopedia of linguistics published in the 3rd millennium. And there is there a great chapter, Linguistic Genetics. And what I will tell you is about what I read in this chapter, written by Wolfgang Reibel. He began to publish in this in 1993, and the encyclopedia is published in 2001. Then, third, the semiotic approach, another huge enterprise, mainly in the United States. I have to tell you, the field of biosemiotics, dealing mainly with biological cell, is born in the United States. Then, genomics and the Human Genome Project, where our colleague Professor Israel was one of the main actors. And I read in this respect 2 papers by Richard Karp about the mathematics involved in this project. And then fields, DNA computing, Adleman 94, Tom Head 87, Adleman the experimental part, Tom had the theoretical part, he is chronologically. Paul Rosenberg Salova, 98, is a book, and there are periodical congresses in the United States about DNA computing. They continue now too. Membrane computing, initiated by my colleague George Paun, and book published by Springer, 2002. Gene assembly in ciliates, initiated by Andrew Ehrenfeucht, he is here in the United States, Tero Harju Petre in Finland, Prescott, a biologist here in the United States, Rosenberg, both in Holland and in the United States, computation in living cells, gene assembly in ciliates. They book at Springer, 2003. Then, another field, matrix genetics. This is a Moscow school of mathematical genetics. His leader, Sergei Pektukov. I will refer to one of his works in 2006. Then, I collect, because there is an inflation now of international conferences on biotechnology, a lot of things having the prefix bio. And with many engineers, many physicists, any kind of professional fields. And for instance, I collected the keywords and the references from an international symposium that took place in Bucharest at the International Center of Biodynamics. And you will see, I have a way to tell you about a field, giving two kinds of information. Some keywords and the journals where they publish. Then I put the field of cellular automata, systems biology, and then two fields which I could name there the metaphorical approach to the biological cell. These are the genetic algorithms and Richard Dawkins' approach based on the analogy between genes and memes. Then, in reference to the protein folding problem, one of the basic unsolved problems related to biological cell, the computational geometry approach, and as a matter of fact, what I told you there, I think if somebody will not bring another information, this was a pioneering paper in both biocomputing, computational biology, and in the field of picture grammars, because it was a way to work with grammars of some pictures, where elements are no longer elements of an alphabet in a linear case, but basic elements of the alphabet are b-dimensional entities. And then, the last one I will consider, the approach to biological cell based on finite and infinite words. And I have to tell you that already just 10 years ago, it was published an important article in Proceedings of the National Academy of Science in Washington by 4 authors. I will give you about the type of regularity they called quasi-periodicity, which simulates the DNA. So here I will not. I will make a selection. No, I am not. I don't have time to speak about all of these things. I will speak about things which are, I suppose, are not known. You don't know them. The linguistic approach, the semiotic approach, a little about genomics to show its interference with the linguistic approach, then about the matrix genetics, a little 0.9, and then I will move to genetic algorithms, to Richard Dawkins, and to infinite and finite words. I don't know what time I will have at my disposal for these things. You see, to understand the degree of loneliness of each of these approaches, I could tell you that you may say, well, I don't know nothing about linguistics and genetics because I am not a linguist. But most of linguists, if you ask them about linguistics and genetics, they know nothing about this. Because in linguistics, like in biology, is the same lack of global knowledge. You know, my lecture here, this lecture is mainly not to, I would say, what I tell you about the biological cell is a symptom of the degree of crisis we are now in our capability to keep under control what happens in the topic we are interested in. The main slogan of this approach, well, before giving the slogan, I will give you a selection of the journals where papers in the field of linguistics and genetics are published. This is very interesting. I am curious if you know these journals. Well, some of them are very known, general biochimie, this is in French, biosystems, cell, child development, chromosoma, cognition, cognitive science, computer applications in the biosciences, current opinion in cell biology, disorders and communication, European Journal of Disorders and Communication, European Journal of Human Genetics, EMBO Journal, Genetica in Russian, Genomics, Journal of Human Evolution, Journal of Speech and Hearing Research, Nature, Journal of Theoretical Biology, this is a very known journal. Nature Genetics, Nature, the huge journal. Nucleic Acids Research, Proceedings of the National Academy of Sciences. You see, there are some journals like this, Proceedings of Nature, which are of very general audience, but they are a minority. Most of the journals, or like Scientific American, obviously everybody knows, but most of journals are not known for many of us. You may understand even the way they make the history of the field is very interesting. The written language as metaphor starts with the early atomists and continues with Aristotle's metaphysics. The variety of the visible world follows from the differences between atoms and from their combinatorial capacity in the same way in which the potentially infinite variety of the Greek text follows from the difference between the 20 letters of the Greek alphabet and from their combinatorial capacity. The same metaphor in biology. Look, now it's very interesting. In 1869, Friedrich Michel discovered the existence of nucleic acid in the center of living cells. So the nucleic acids are known for a long time. In 1893, Michel claims that the relation holding between the letters of the alphabet and the huge number of words obtained from them could explain the relationship between the information contained in the nucleic acids of our cells and the variety of life forms. You see? This is just a premonitory idea. In 1893, you cannot write the history of biocomputing by ignoring such a statement. Look, das alle Reichtum und alle Mannigfaltigkeit erbringen, Übertragungen ebenso gut darin ihren Ausdruck finden können, als die Worte und Begriffe aller Sprachen in den 24 bis Buchstaben der Alphabet. Friedrich Michel, this is exactly quotation. In 1943 then, Erwin Schrödinger, What is Life, suggests a genetic alphabet similar to the Morse code. Mistakes in the process of reading and copying the code lead to mutations. Oswald Avery, 1944, the nucleic acids and not the proteins contain the genetic information necessary to unfold the functions of a pneumonic bacterium. These facts makes Erwin Chargaff to propose the idea of a grammar of biology. So you always, metaphors precede models. All these proposals were confirmed in 1953, Crick Watson, the long strands of DNA, Schrödinger's punch Morse tapes, have the structure of a double helix. The metaphor of language in the form of alphabetic script is generally accepted in molecular biology. In the USA, the National Center for Biotechnology is running a large database called Medline. Until 1997, this base had a subset in molecular biology, genetics, comprising some 700,000 full text documents, starting from about 1966, and in 1998 this subset was merged with the general biological database, the resulting whole containing now some 9 million full text documents. In this database permits searching for any expression. It is easy to demonstrate the presence of the language and the script metaphor in the whole range of text in molecular biology. Recently, a new metaphor is being used more and more. The genome is like an encyclopedia. Dictionary is still relatively rare. Part of this encyclopedia may be formatted in a different way, like bold or italics. Structural similarity as the deeper reason behind the language metaphor. This is a deeper relationship between the way the grammar of biology and the grammar of natural language are working. In both cases, information is coded into a linear sequence of basic elements, nucleotides in one, phonemes or letters in the other case. Since this code sequence of elements is of necessity linear, both systems have to cope with the same fundamental problem. And this is the following. How can a one-dimensional medium transmit the information required for the construction of 3 or even more dimensional entities? The complexity of the information to be transmitted becomes evident when we imagine that all of us originated from one single egg cell whose genome was replicated billions of times by successive divisions of cells, so-called mitosis. The final result of this evolution is a well-structured 3-dimensional body. The same kind of process takes place in human speech. What we want to utter are complex ideas and representations. Those ideas are broken down into sections encompassing a series of subsections made out of paragraphs. The paragraphs themselves consisting of linked sentences and propositions. Sentences are again broken down into entities called clauses, phrases, words, and still linear sequences of words consist of letters. Also, the first part of this process, the breaking down of the complex, represents or the complex representation into successive hierarchical layers of pieces, resulting eventually in a linearized sequence of basic elements. Does not have its counterpart in genetics. The subsequent reconstruction by the reader or hearer does indeed, in order to achieve these genetic processes, rely on principles strikingly similar to those holding in language. Well, I don't have time to follow all this, but then we have, you see, similarity. Ultimately, they claim here is that heredity is essentially language, and in many respects it is like the natural language. Like in language, genetic reading processes tend to be highly reliable, thanks, among other things, to a considerable amount of redundancy in genetic information. Now, similarity concerning the basic principle in both language and molecular genetics. The principles allowing the reconstruction of multidimensional wholes from linear sequences of basic elements are identical. Double articulation, different classes of signs, hierarchy, combinatorial rules on the different levels of hierarchy, and linking the principle of hierarchy and combinatorial rules. Walls are always more than the sum of their parts. This is very important. In linguistics, double articulation comes from André Martinet in the domain of life. A similar principle comes from Émile Boutroux, 1875. But you see, the symptom of today's culture. Linguists ignore Boutroux and biologists ignore Martinet. As have science in language, DNA and proteins gave two different functions. I will avoid these details. You may have a general, you see then, the existence of two totally different kinds of genes and as a consequence of proteins translated from these genes. Proteins that give the cell its special shape and its typical metabolic functions, and proteins whose task is to regulate the reading of other genes. The same kind of regulatory processes are necessary for the functioning of language. And here is a description of this thing. I no longer have time to tell you here. Now, what is similar and what is different in the linguistic system and in the genetic system? They have to cope with the same basic problem, to construct polydimensionality out of a linearized code. But the sender-receiver duality, so important in language, does not exist in genetics. Second, cellular processes are energetically optimized by evolution. So in genetics, the potential for economically induced change in an already economically optimized system is minimal. By contrast, in human language and communication, economy is a major factor and one of the inevitable activators of language change. C. Both systems have double articulation. The second level, the genetic system, makes use of 20-odd amino acids, where the natural language uses their word signs. The difference is decisive. In the genetic system, there exist 4 power 3, 64 possible triplets of the 4 letters, A, T, G, C, to which correspond only 20 odd amino acids. So, synonymy is strong in genetics, whereas true synonymy is extremely rare in natural language. True synonymy. Well, some of these claims can be, we can talk about them. Polysemy, on the other hand, polysemy and even homonymy are frequent in natural language and generate the problem of syntactic and or semantic context for disambiguation, but very reduced in genetics. Here, opinions don't agree always, because already in the genetic dictionary, there are homonymy phenomena, many homonymy phenomena. D, the result of gene transcription and expression is something material and palpable, as in the case of our bodies, whereas the result of the reading process, for instance, in the case of a novel, rests something highly immaterial and volatile. subject to interpretation by different readers. But genetically inherited properties, like behavior, may also have such ambiguity, while reading a patent specification leads to concrete things. And E, last two differences. In genetics, the same text exists up to billions of times in one and the same body, because the genetic information is repeated in each cell separately, and it is read very selectively in any cell. On the other hand, the regulated elements in text of natural language facilitates the reconstruction of a sense out of linearly scattered elements, needing a mostly linear reading of the text. Regulatory proteins are designed to make possible a specific and selective reading of selected genes in one cell, such that, nevertheless, the aggregation of individual cells forms an ordered and functioning whole, which is far from more than the sum of its parts. But printed text increasingly enables selective reading techniques too. And the last one, the primary aim of genetics is high fidelity replication. On the other hand, text in natural language may express whatever comes into our minds according to our creativity. But evolution shows that there is another kind of potential creativity in the genetic code. It allows for mutations due largely to flaws in the reading process. So, two possible metaphors for the genome: the computer program and the encyclopedia. The activation of RNA polymerase on a certain gene follows exactly the pattern of a computer program. If activated, and well, I don't have time to develop more this. Are biologists aware of the structural similarities? Most researchers in microbiology are familiar with most of the metaphors outlined above, but seldom are they aware of the basic problem posed by the linearity of DNA code as opposed to the 3 or even 4 dimensions of the resulting phenotype. Detecting irregularities in DNA by linguistic methods, deciphering human genome. Unfortunately, I no longer have time to, because an example of grammatical approach. Oh, look, you see, it is very interesting that in the Human Genome Project, it seems that the grammatical approach is absent. But people working in linguistic genetics, they claim that grammars may have some relevance here, but they are too technical. I can no longer go in this in these details. Well, I will. I have to move to. No, I no longer have time to. I can show you to see the hidden Markov models, algorithms shaped according to the models of context grammars, turn out to be powerful tools for the detection of so-called homologs, which is their claim. Many approaches aim at finding the entire regulation process of a gene, it seems to me that this total lack of communication between people working in this field and people working at the Human Genome Project is something negative. All these approaches showed indirectly that the genetic system has the properties of a true language with its grammar or grammatical regularities, the type of rewriting rules leading to dyad symmetry in the above example was even used by Chomsky in order. Yes. Nevertheless, one basic problem persists. These algorithms are essentially bottom-up models holding for cells as the most for cell assemblies or cell compounds. The complementary top-down component necessary to understand the processes of embryogenesis with these hierarchical levels and corresponding categories still remains in the dark or at the best in twilight. Now that the human genome has been successfully deciphered, the true complexity of the grammar of biology will become visible. And then there are examples of metaphorical transfers from chemistry to linguistics. I evoke here the case of Lucien Tenière, who using the idea of valency. Valency is from chemistry. And we have the valence grammar, also a type of generative grammars. What is here? Molecular grammar consisting of the rules that govern the assembling of molecular units into messengers and their higher order structures, such as hormones, DNA binding proteins, or transcription complexes, was considered by Ratner. This is a Russian author. Biosystems. Biosystems are able to discover in the genetic system nearly all the features attributed to human language. By Charles Hockett, The Origin of Speech in Scientific American. If at the most 5 to 10% of the DNA contained in a genome is represented by genes, what is the function of the rest? Conjecture. 90 to 95% of DNA could incorporate spatiotemporal genes, with their function being the control of the folding patterns or conformational states of DNA and the topology of chromosomes. The non-coding part of DNA in eukaryotic genomes are thought to encode a language which programs organismal growth and development. So, this was only about the linguistic approach, and I promised you to tell a lot of other things. For instance, do you know what does it mean, biosemiotics? They also claim their main aim is to understand the functioning of the biological cell. Well, I will give you first the journals where they publish. Biosemiotics, some journals. This is one of the huge American enterprises in the field. Biosystems. Annals of the New York Academy of Science, Boston Studies in the Philosophy of Science, Astrophysics and Space Science, American Journal of Semiotics, American Scientist, Animal Cognition, Annual Review of Neuroscience, Communication and Cognition, Cognition and Brain Theory, Cognition and the Symbolic Process, Comprehensive Biochemistry, cybernetics and human knowing, cosmos and history, developmental biology supplement, ecological complexity, ecological modeling, experiencia, entropy, Journal of Biosemiotics, Journal of Consciousness Studies, General Systems Bulletin, Journal of Atmospheric Sciences, Journal of Literary Semantics, Journal of Philosophy, Journal of Theoretical Biology, International Journal of Applied Semiotics, International Journal of Computing Anticipatory Systems. And continuation, because it is a huge enterprise, biosemiotics, other International Journal of General Systems, Language, Mathematical and Computer Modeling, New York Review of Books, Physical Chemistry, Chemical Physics, Physical Review Letters, Physics Today, Perspective in Biology and Medicine, Philosophy of Science, Proceedings of the American Philosophical Society, Psychiatry, Proceedings of the National Academy of Science. You see, Rivista di Biologia Biology Forum, Science, Semiosis, Evolution, Energy, Science System Studies, Semiotica, the Nordic Journal of Philosophy. Keywords in the field, keywords in the biosemiotics. Organic information. You see, this is a huge problem. Is the DNA, what we call the DNA information, the only type of organic information, or are there some other? The claim is that there are many other types of organic information. Organic sign, another key concept, because the claim is that life is ultimately, this is a claim, life is ultimately a sign process, not behind biology, physics, chemistry, you have this common denominator, sign process. Organic meaning, organic codes, ribotype, physicalism, manufactured molecules, semiosis, interpretation, operative definitions, spontaneous molecules. These are some of the keywords in the field. And genes and proteins differ from all the other molecules, not because of their size, shape, or chemical composition, but because they are produced in a totally different way in the inorganic world. The structure of molecules is determined by some internal factors, the bonds existing between their atoms. By contrast, in living systems, genes are built by molecular machines which physically stick their nucleotides together according to the order of that template, which is eternal to the growing molecule. In a similar way, proteins are made by molecular machines which bind amino acids in the order prescribed by an external template of nucleotides. So, the structure of molecules is determined by internal factors in the inorganic world, external factors in the living universe. We have also natural, spontaneous molecules in the inorganic world, artificial, manufactured molecules in the living world. In the history of life, molecular coffee came into being when the first coffee makers appeared on the primitive earth and start making copies of nucleic acids. This means that natural nucleic acids had already been formed by spontaneous reactions on our planet, but that was no sign of evolution. Only the coping of genes could ensure their survival and have long-term effects, So it was really the arrival of copy making that set in motion the chain of processes that we call evolution. The first major transition of the history of life, Maynard Smith's 95, is described as the origin of genes. This means of the first molecular machines making copies of nucleic acids. The origin of proteins. Unlike genes, proteins cannot be their own templates. One cannot get proteins by copying other proteins. The information for manufacturing proteins comes from genes. The important feature of the protein makers was the ability to ensure a one-to-one correspondence between genes and proteins, otherwise it would be no biological specificity. This means no heredity and no reproduction. Life would not exist. Protein synthesis arose from the integration of two different processes, and the final machine was a code and template dependent peptide maker. A code maker, the second major transition of the history of life, is described as the origin of proteins, here of code makers and of code machines and code makers. The first molecular machines which added coding and populated the Earth with proteins. And so the difference in respect to what is known, the cell as a trinity and the emergence of the first cell. Beginning with Johansen, life is considered a duality of genotype and phenotype. This representation was confirmed in the period 40-60, when it became clear that genes carry linear information, whereas proteins function by their three-dimensional structure. The gene-protein duality is equivalent to other dualities: heredity, metabolism, information, energy. The information-based biology of the past century is in contrast with the energy-based biology of the 19th century. After learning in the 1950 and 1960 that genes and proteins are not formed spontaneously in the cell, but are manufactured by a system of molecular machines based on RNAs, we learned that the components of the manufacturing system were called by Marcello Barbieri. This is the author of a basic book, Introduction to Biosemiotics, ribozoids, while the system itself was given the collective name of ribotype. So the cell appears in this way as formed of genes, proteins, and ribozoids, and as a trinity of genotype, phenotype, and ribotype. Genotype, the seat of heredity; phenotype, the seat of metabolism; ribotype, the seat of genetic code, the code maker of the cell. Science and meaning exist only when a code maker creates a correspondence between two distinct entities. The code maker is the agent. Science and meanings are the instruments of semiosis, and they are code maker dependent entities. Science and meanings exist as protein synthesis because there are code makers dependent entities in that process, and the genetic code is a correspondence between those entities. How many organic codes? This is a big question. According to modern biology, the genetic code is the only code that exists in the organic world. Is this true? In order to answer this question, let's start with the difference between coding and coding, visible in transcription and translation. In transcription, an RNA sequence is assembled from the linear information of a DNA sequence. And in case of a normal biological catalyst, an RNA polymerase is sufficient because each step requires a single recognition process. In translation instead, two independent recognition processes must be performed at each step, and the system that performs the reactions, the ribosome, needs special molecules, first called adapters, and then transfers RNA in order to associate codons to amino acids by means of the genetic code. In its absence, we would lose biological specificity. These things can be generalized. We are used to think that biochemical processes are all catalyzed reactions. But the difference that exists between coding and coding tells us that we should distinguish between catalyzed and codified reactions. In the first case, only one recognition process appears at its step, while in the codified reactions, two independent recognition processes at each step are necessary, and we need a set of coding rules. The catalyzed reaction requires catalysts, the codified reactions need adapters. This means catalyst plus a code. An organic code is just a set of rules of correspondences between two independent words, such that there is a system of adapters and a set of rules. So they give a list of other organic code other than the, the splicing code, the signal transduction codes, the compartment codes, the cytoskeleton codes, the sequence codes, the adhesive codes, the sugar codes, the histone code. Organic information, this is another basic issue still in controversy. Because the idea of information promoted by Watson Crick was that the linear sequence of nucleotides represents the information carried by a gene. A few years later, the mechanism of the protein synthesis was discovered, is the transfer of linear information from genes to proteins. Physical stasis. So this is no longer accepted. The definition of information by Crick and Watson and Crick is no longer accepted by biologists. Physical stasis, heredity, and organic information are ultimately physical. This would imply no essential difference between life and inanimate matter. This is rejected. Three different.
Well,
yeah.
We need to.
Here is the three different semiotic models of the biological cell. It was first model by Marcel Florkit, defined the biological system as a duality of genotype and phenotype. Second, Thomas Sidrock adopted the triadic scheme of Charles Sanders Peirce. There can be no semiotics in absence of interpretation. But then, in 81, 85, 2007, Barbieri considers that the necessary and sufficient condition to have a semiosis is to have a coding process, not an interpretation. Well, and this is a trial he proposed. Is the cell a semiotic system? Some at left, those who answer yes. Modern biology says no, but biosemiotics says yes. Keywords, sign, code, and meaning. Arguments for no. Sequences and codes are only metaphors and not fundamental entities. They can be reduced to physical entities. Arguments for yes, only formation of spontaneous molecules can be described by physical quantities. Molecular artifacts like gene proteins require new basic entities such as organic information, organic sign, organic meaning defined by operative definitions as objective and reproducible as objective as physical quantities. Semiosis is a result of interpretation, claims, those who say no, and you have no interpretation in the genetic code, and the author says, no, we did no interpretation, it's enough to have coding. And I will stop here. Unfortunately, as you see, I was not able to cover all I promised you at the beginning. Maybe if you accept, we continue after a break.
Okay, let's take a break. Thank you very much.